What's up, Tumnad lectures? In this video, we are looking at what you call the basics of what you call the proteins. If it's your very first time to watch me, please make sure you like the video, comment down in the comment section, and then please subscribe. Let's go ahead and get started. Before we go into the detail of chemical digestion and absorption of the proteins in the next video, today we want to take a little of the time and understand the basics of what we call the proteins before we go into what we call the details of chemical digestion and absorption. Because it makes a lot of sense to first know the idea of the chemical structure of what we call the proteins. Because we need first to know what are those kind of the molecules that make up what we call the protein. What is that type of the bond that joins these kind of the macromolecules together to make what we call the protein. Okay. Today we are looking at what we call the basics. Okay. We have come up with this kind of this big structure which we are calling an egg. It's very nice, delicious, very important to enjoy. The question is, which kind of the macromolecule are we going to be getting from what we call an egg? It is super very important because this egg is going to be containing one of the most important macromolecule which we call the protein, and it's going to be our discussion of today's video. Okay. Now, when we zoom on this kind of the protein we are getting from this kind of an egg, we are going to be getting this kind of the long chain of the polypeptide which make up what we call the protein. Okay. So now, in other words, a protein is going to be a long chain of the polypeptide which is going to be linked by one of the most special type of the bond which I'm going to talk about. Okay. Now, when you see these very many kind of these dots, red, green, black, blue, these ones, they are showing us the amino acids, okay? Because the amino acids, they are the building blocks. They are the building blocks of what we call the proteins, okay? So now when we talk of amino acids, these are the building blocks of what we call the proteins. They are the absorptive kind of the proteins that the body requires, okay? Now, we want to take a little bit of this single amino acid and we try to zoom in and see how it looks like on the chemical structure at a molecular level. Okay? Because it's very necessary. Now, besides that, on every end of what we call the amino acid, we are also going to be having what we call a carboxyl end and what we call the amino end. That is super basic, okay? On every kind of the amino acid or even a very long of what we call the polypeptide, we shall have a carboxyl end and then an amino end. That is it, okay? Now, looking at the chemical structure of what we call the amino acid. Now, we have a lot of the amino acids. The body contains approximately around 20 different amino acids. Okay, today we want to see one amino acid and we see what is that kind of the part that differentiates the amino acids from one another, because there are 20 of them which are a bit different. Okay, now this structure here shows amino acid, one individual amino acid. So now let's go and see what are some of those kind of the atoms, the kind of the elements that really make what we call an amino acid. We are going to be having what we call the carbon. This is going to be what we call the carbon. You're going to be having the oxygen. We're having the hydrogen. We are having the nitrogen. So we are having a lot of the elements that are coming together to form what we call the amino acid. Same like what we call the carbohydrates, with the exception that we are not having the nitrogen in what we call the carbohydrates. It's all that simple, okay? So now, let's go ahead and see the different parts that make what we call the amino acid. The first one is going to be this kind of this structure here. Super very important. Okay, this kind of this structure here is what we call the amino group. Okay, is what we call the amino. Is what we call the amino group. Okay, and then when we come to this kind of this carbon here, this one is what we call the central alpha carbon. Okay, this one here is what we call the central. Is what we call the central alpha. The central alpha carbon. Super very important. Okay, and then when we come on this kind of this structure here, this one here, the whole of this structure, super very important. This one here is what we call the carboxyl group. Okay, this one here is what we call the carboxyl. Is what we call the carboxyl group. You may find some other people writing it as the carboxylic acid. Okay, this is going to be the acidic end of what we call the amino acid. And that is why this one is called the amino acid, just because we have the amine group and then the acidic group, which is the carboxyl group. And that's why the name is the amino acid. Okay. And then when we come on this this structure here, this one super very important. This one is what we call the variable chain. Okay, this one is what we call a variable. Is what we call a variable chain. Okay, and now this variable arrow chain is the one that is going to be differentiating amino acids from one another. Okay, all amino acids are going to be having the amino end. They are going to be having what we call the carboxyl end. They have what we call the central alpha carbon. They also have what we call what we call hydrogen atom. Okay, this one here is what we call the hydrogen. Is what we call the hydrogen atom. Okay, they'll have all these structures. What will differentiate them from one another is this kind of the side chain of what we call the variable chain. It's all that simple. Okay, so now when you see the difference between the different amino acids, you have to be checking on this kind of the other variable side chain. Okay. Now, having seen the different parts that make up the amino acids, let's go ahead and see how are these amino acids joined to one another. What happens if I react one amino acid to another? How are we going to be forming one of the beautiful bonds which I'm about to talk about? Okay. Now, this one here is going to be one amino acid, and this one here is going to be another amino acid. Let us call that this one is going to be amino acid A. Okay, this is going to be amino acid A, and this one will be amino acid B. These amino acids are going to be different. Okay, but we want to see what happens when these kind of the amino acids come together. Okay, we want to go ahead and see what happens when these amino acids come together. Okay, now when these kind of the amino acids come together, when they react one another, we have a specific type of the process which is going to be happening. When you come on this carboxyl end of this amino acid A, and you come on amino end of this kind of amino acid B, we are going to be seeing a very important kind of the reaction. We are going to be seeing this kind of the hydroxyl ion, reacting with what we call the hydrogen ion on this another kind of the amine end of this kind of the amino acid B. Okay, we are seeing the hydroxyl ion of amino acid A reacting with the hydrogen ions on what we call amino acid B. Guess what are we going to be forming? When these two kind of the ions react together, we are going to be forming what we call the water. Okay, now by doing that, what are we going to be forming? We are going to be forming one of the most strongest kind of the strong covalent bond ever that's going to be linking these kind of amino acids together. We are losing water, but we are forming one of the strongest covalent bond. This bond is going to be coming on what we call the carbon on what we call the carboxylic end, and also what we call the nitrogen, what we call the amino end. This kind of the bond, which has been created here, following the loss of water, is a special type of the bond. You know how this bond is called? This bond here, which has been created, is what we call a peptide. Bond. Okay, is what we call a peptide bond. It's all that simple. Okay, now what happens is that the formation of what we call peptide bond. How do we call that kind of the chemical reaction that leads to the formation of what we call the peptide bond? That kind of the chemical reaction that leads to the formation of what we call the peptide bond is what we call dehydration synthesis. Okay, is what we call dehydration synthesis. Okay, that is the reaction which is going to be taking place by the reaction between amino acid A and amino acid B, leading to the formation of what we call the peptide bond, following even also the loss of what we call water. Okay, it's all that simple. Now we have now joined the amino acid A and amino acid B. Guess what have we formed? We have formed what we call a dipeptide, because now they are now two amino acids joined together, and that's what we call a dipeptide. Okay, so now we have formed this one here, which we call a dipeptide. Okay, it's all that simple, meaning that if we join another amino acid and they become three, it will now be a tripeptide. It's a matter of case of joining more of these kind of amino acids, which will end up having a long chain of amino acid, which we call a polypeptide. It's all that simple. Okay, now what happens is that if we are to break this kind of the proteins during chemical digestion, we are going to be targeting this bond. Okay, during chemical digestion of the proteins, we are mainly going to be targeting this kind of the bond, which we are calling the peptide bond. It's all that simple. Now, what is going to be happening during chemical digestion? One of the most important kind of the process that is going to be leading to the breakdown of what we call the proteins, or what we call the peptide bond, is going to be if you add this water back on this kind of the dipeptide, this bond is going to be breaking. Okay, meaning that one of the most important kind of the process during chemical digestion of what we call the proteins is going to be addition of water. How do we call that process? Hydrolysis. Okay, so now if we want to get back to individual amino acids, we are going to be adding water. So now this process, which is going to be now converting back the dipeptide back into the individual amino acid, is going to be involving addition of water, which we have called hydrolysis. Okay, we have called it as what we call hydrolysis. Okay, so now hydrolysis, when we add water on this dipeptide or even a polypeptide, we are going to be forming individual kind of the small portions of what we call the proteins. However, this kind of the hydrolysis only will take a lot and a lot of time to break even this kind of the single peptide bond. What are we trying to mean? We need a specific type of the enzyme that is going to be speeding up this hydrolysis process to make sure that these kind of the peptide bonds are broken within the shortest time possible. Because if you leave hydrolysis only, it will take years to break even one single peptide bond. It is such a beautiful thing. So now we need a lot of enzymes that are going to be catalyzing the hydrolysis process to make sure that peptide bonds are broken within seconds or even what we call minutes. Now the question is, how do we call those kind of the enzymes that are going to be speeding up the hydrolysis process to make sure that the peptide bonds are broken down within the shortest time possible? This hydrolysis process is going to be catalyzed by what we call proteases. Okay, we have what we call pepsin, and even also what we call pancreatic proteases. Okay, pancreatic proteases. Okay, these are the two main types of the enzymes that are going to be catalyzing the hydrolysis process to make sure that the peptide bonds are chemically broken down into smaller chains of what we call the proteins, which can easily get absorbed into what we call the enterocytes. Okay, the key point to note is hydrolysis only cannot break the peptide bonds. It will take a lot and a lot of years to break this peptide bond. However, pepsin and then other pancreatic proteases will come in to catalyze this hydrolysis process, leading to the formation of what we call the individual amino acids. Okay, it's all that simple. So now, to Munad, I have talked a lot of things. I hope you have enjoyed the video. If you have enjoyed it, please, you know what to do. Hit the like button, comment down in the comment section, and please subscribe. Thank you very much for watching, to Munad, as always. Until we meet next time.